Hello, good afternoon, everyone. I hope you guys had a restful week last week. Have a nice break, recharge a little bit, and we are back at it again. So today's tutorial, we will be taking up the Test 1 review package, which you can find on eCentennial. If you haven't already downloaded, that's okay. You can just follow along as I take it up, write down in your notebook or something, and then you can download a copy afterwards. Okay, so the test, of course, is going to be on the Thursday. It's going to be available for the whole day from 6 a.m. to 11:59 p.m. And you can find all the details on eCentennial. So let's get started. So the first question, it's related to lecture 2. What holds the atoms together in a water molecule? How would you describe a water molecule? What interactions exist between two adjacent water molecules? Okay, so I can draw a water molecule. There's an O here, there's an H and H. All right. So what kind of bond exists between the oxygen atom and the hydrogen atom? Can anybody tell me? What is the bond between oxygen and hydrogen? That's the question. Yep, Moira said this is a covalent bond. That is correct. So in covalent bond, we have a sharing of electrons. Sharing of electrons. Now, this sharing is not necessarily going to be an even sharing. Okay, we have one atom potentially pulling the electrons toward itself a little bit more, and that is the oxygen. Okay, for that reason, the oxygen is going to end up being slightly negative. Okay, and the hydrogens will be slightly positive. Right? So overall, there is an uneven distribution of electrons in the molecule. Okay, so uneven distribution. Let me put some lines here so it's easier to write. Uneven distribution of electrons in H2O, right? And so that makes it polar. It is a polar molecule. However, there is no excess electrons or deficits of electron, right? Nobody gave for the water molecule extra electron. No one took away any electrons. For that reason, even though it is polar, the entire molecule is still neutral. Okay, so no excess or deficit of electron. So it is neutral. How would you describe the water molecule? Polar and neutral. What holds the atom together? Covalent bond. Now let's take a look at what happens when you have two water molecules right next to each other. Let's say we have an H2O here, and then I'm just going to put another H2O here. And maybe just for good measure, let's just throw another one in. Right? So what happens is the slightly negative oxygen of one water molecule will be attracted to the slightly positive hydrogen of another atom. Okay, so this interaction that exists between water molecules, what do we call that? Can anybody tell me? What do we call these pink dots that I'm trying to represent here? Adriana said hydrogen bonding. That's correct. So these are hydrogen bonds. Okay. Not ionic bond. Ionic bond is when you have a metal and a non-metal that connects together, like a sodium and a chlorine come together, then you have an ionic bond, right? These are interactions between molecules rather than within a molecule. Okay, next question. If you supply radioactive nitrogen atoms, so something that you can trace that you can detect. If you supply that to a cell and let it grow, what molecules will you expect to find the radioactive labels in? So this is basically, another way of asking you what macromolecule has nitrogens. So let's just write them all out. We have carbohydrates. Carbohydrates is made up of carbon, hydrogen, and oxygen atoms. What about proteins? Okay. What are the elements that we can find in proteins? Anybody? No? Excuse me. There's carbon, there is hydrogen, there's oxygen. It's going to be nitrogen as well. And then a trace of sulfur, a little bit of sulfur. Lipids. Lipids, we have carbon, hydrogen, and oxygen. And for nucleic acid, we have CHO and as well as phosphorus. So if you supply radioactive nitrogen to a cell and let it grow, you will find the radioactive nitrogen in the proteins that it makes as well as the nucleic acid. Okay? So that's the answer, proteins and nucleic acid. Next question. Regarding DNA, what is the monomer of DNA? Okay, so monomer of DNA is nucleotide. Can anyone tell me what are the 3 parts of a nucleotide? What parts will you find in a nucleotide? Sakina said sugar. That's correct. So there is going to be a sugar. And for DNA, it is a deoxyribose sugar. Adriana said phosphate group. That is also correct. Okay, phosphate group of PO4 3-, right? And a nitrogenous base. That's correct. All right. So this is a sugar. It's a ribose sugar, but it's missing an oxygen. That's why it's called deoxy. Phosphate group PO4 3-. Nitrogenous base for DNA can be A, C, T, or G. Okay. How are the nitrogenous base classified? So this is A, B. What are the 2 categories of nitrogenous base? Anybody know what I'm talking about here? Nitrogenous base can either be, that's correct. Adriana said pyrimidines. Pyrimidines or purines, right? You should know which one is which. Purines are adenine and guanine, right? Pure as gold. That's how you can remember it. I told you that before. And pyrimidine is thymine and cytosine. You should also know pyrimidines is single ring. Okay, 1 ring structure. I'm just going to draw it out for you so you can visualize it. Right? So there's a single ring in the structure. Whereas purines, they are going to have double ring in its structure. Okay. There we go. So 1 ring, 2 rings. Okay. Purine has 2 syllables, 2 rings. That's how you can remember it. Okay. And pyrimidine, 1 ring. Double ring, we'll say. Double ring. or like two fused rings. All right. Next question. What is the base pairing rule for DNA? Base pairing rule is this. Okay, let's say I have one strand here and another strand. If this is, Alex asking me to scroll up. Sure. Like here or like even higher? Oh, first question. This one? All right, no problem. Base pairing rule. What is the base pairing rule? Well, let's say we have A, C, G, and T on one side. Then the other side is going to be T, G, C, and A, right? Okay, so this is the base pairing rule. A always pairs with T, C always pairs with G, which is, another way to say this is pyrimidine. A pyrimidine will always pair with a purine, right? And then I think later on there is a question that asks, what is the significance of the base pairing rule? So by having a pyrimidine pairing with purine, you ensure that the width of the DNA is always going to be 3 rings wide. Okay, imagine a ladder, right? You want the distance between the two, I don't know what you call them, like the handles, right? Where you put your hands on. You want the rucks to be the same length, right? It would be weird to have a ladder that's like, you know, some part is longer and then some part is shorter, right? Like that doesn't make any sense, right? So in our DNA structure, we want to ensure that the width is going to be consistent along the entire length of the DNA, right? So this ensures the width of the DNA molecule remains constant along its length. Does that make sense? Yes, I will post this sheet after class, as I always do. But are you everybody with me? You understand what I'm saying? Like three rings across? I hope so. So the pyrimidines, right? One ring. So there's a one ring here, one ring here, one ring here, one ring here. And then for our purines, 2 rings, 2 rings, 2 rings, 2 rings. So across is always 3 rings. By the way, just so that you know, right? Like this review is generated based on the test, right? Okay, so like I created a test for myself randomly, just like how your test is being created, okay? And then I created these questions based on what I see on my randomized test, okay? So a lot of these concepts are, I wouldn't be surprised if it shows up on your version of a test as well. So make sure you pay attention. I should also mention someone, like actually a few people emailed me, asked me if this is the only thing we should study for to prepare for the test. Absolutely not. Okay, this is just it's a good starting point, but you know, all the study guides that we did in the past, all the lecture notes, those are going to be fair game as well. So you have to make sure you study from those as well. Did I miss anything? Yes. What force holds the nitrogen bases together along the length of the DNA molecule? All right. So what's holding these molecules together? These are nitrogen base together. Well, we're gonna have some hydrogen bonds again, right? So for A and T, there are 2 hydrogen bonds, and then for C and G, there's gonna be 3 hydrogen bonds. Okay. So 2 hydrogen bonds between A and T. 3 hydrogen bonds. Hydrogen, sorry, I'm missing the word bond. let's try that again. 2 hydrogen bonds between A and T, 3 hydrogen bonds between C and G. So if you have a strand that has a lot of A and T's, it's easier to pull them apart because it's less interactions between them, right? Compared to something that has a lot of C and G. I think that is it. That is it for number 3. Does anybody have any question? No? All right. Number 4. What is dehydration synthesis? Illustrate with a diagram. Illustrate with a diagram the significance of this process in building polymer. Sorry, that doesn't exactly make sense. Illustrate with a diagram to show the significance or something is supposed to be. So here we go. We have, let's say, all these individual building blocks, right? So these are monomers. And what we can do is we can link them together in the process of dehydration synthesis. Another word for dehydration synthesis, as you know, is condensation reaction. So you remove a water molecule from the monomers and you connect them together. You create a covalent bond, right? That links them together. And in the process, you generate some H2O. Right? So over here, if you have 1 connection, 2, 3, 4, 5 connection, then you would have 5 H2O. Right? So each of these bonds, you should know it's a covalent bond. Covalent bond. Right? So this would be a polymer. And if I want to break the polymer, what is the opposite reaction of dehydration synthesis? What do we call this? That's right. It is hydrolysis. Very good. So you will need to add a water. The addition of a water molecule will break the bond and then separate it back into the monomers. Right? So we did the question like this before, but just for good measure, let me ask you again, because it has been a while. Let's say I have something like this, right? A hexagon. You don't really have to know what the hexagon is. I mean, you will learn this in organic chemistry, but each of these corners is a carbon. But let's say there's an OH. There's a question. Are we going to draw this in the test? No drawing. Everything is multiple choice, and you just click. But I could draw stuff for you and ask you what's going on. So for example, I can ask you, what happens if this molecule and this molecule is going to undergo dehydration synthesis? What's the product? Right? What is the product going to be? Right? So you'll be like, oh, okay. I see an OH here. Let's take away the OH. Let's take away the H. And I would have the little hexagon from this molecule. Then it's going to connect to the oxygen over here. And then you will have your pentagon from the other molecule. Okay, so this would be the answer. Right? So essentially, you're going to have a new bond that connects together the rings. Okay, and this would be from this molecule right here, and then you will have this thing over here. Okay, including the O. Does that make sense? If I give you something like this and I ask you to choose which one is the correct product, will you be able to do that? I certainly hope so. All right, moving on. Regarding enzymes, okay. So what is an enzyme? Right. Enzymes are proteins. That's the first thing to know. Okay, they're proteins. So they're made up of amino acids. But exactly what are they? Okay, they catalyze chemical reactions in the body. Okay, by lowering. So this answers both like A and B, right? By lowering the activation energy, right? So let's refresh your memory on what activation energy means. So, you know, this is a graph, and then, you know, people usually get a little bit nervous when they see a graph, but let's just think about this. Let's say this is energy. The higher up you are in the graph, the more energy it has, and this is time, right? So let's say our reactant starts here. Right? Our reactants has so much energy. Everything has energy, right? Like you think about like a chocolate bar that has energy, right? Okay. And then you think of like, I don't know, like nuts or something that has energy. And then the chocolate bar probably has more energy than the nuts, right? Okay. We talk about chemical energy here. And you can always tell, you can look at the calories at the back, right? Okay. So is that same thing for like chemical reactions. If you look at the reactants, they have their own starting amount of energy. It's like sort of like how much calorie it has, right? If you were to eat them, but of course you're not gonna eat stuff in, you know, in chem lab, but that's essentially what it means. Okay, so what we're gonna do is, in order for the reactants to actually react, you need to supply a little bit of energy before it is going to react. Okay, so that extra bit of energy that you need to supply in order to kickstart the reaction, we call that the activation energy. Remember in glycolysis that we learned before the break, in order for you to react, to make the glucose reactive, you must first invest 2 ATP, right? I said, right? It takes some initial investment before you can yield more ATPs in the end, right? So that concept is similar to what we're talking about here, right? You must supply some energy to make the reactants a little bit unstable before it's going to react. The problem is, most of the time, the activation energy is going to be quite high, and it's going to happen very slowly in the body. Okay, so they will still become products, right? But it happens very, very slowly. In the presence of an enzyme, right, what happens is the enzyme will allow this reaction to occur, but with less activation energy. Okay, so over here, the enzyme will lower the activation energy, allowing the reaction to occur faster. Let me use an analogy to help you understand if this is still a little bit confusing. A lot of you may be planning to go into the practical nursing program, right? That's part of the reason why you are in this program. So, you know, to get there, let's say the cutoff is 80%. You must have an average of 80%, right? So not everybody's going to make it. That's just the reality of it, right? Maybe 20% of you will make it if the cutoff is 80%. So that the 80% is like the activation energy, okay? If we have an enzyme, right, then the enzyme will lower that cutoff. Instead of 80%, now 60%. All you need is an average of 60%, right? Then a lot of you are going to be able to make it, right, to the nursing program, right? So it makes it easier for the process to take place. And that's exactly what the enzyme does, okay? It lowers the activation energy. I hope that makes sense. So next question, what factors affect the functionality of an enzyme? Okay, so hit me, guys. Just list a bunch of things that you think is going to affect the functionality of the enzyme. What question is this anyway? This is C. Okay, please list them and I'll write it here. Temperature is definitely going to affect it, right? So temperature is going to disrupt the secondary structure, tertiary structure, right? And the primary structure, right? Sorry, and the tertiary structure, but not the primary structure. pH is also correct. Very good. What else? I'm looking for a few more. What if this is a multi-select question? I ask you to select everything. That concentration of what though? Okay, yeah, someone said charge ions. Yeah, so presence of ionic molecules, right? In the surrounding, in the solution, that could affect it. Okay, what else? What else? Anyone else? Okay, type of solution, right? Is it hydrophobic? Is it hydrophilic, right? If it's hydrophobic, it will cause your enzyme to flip inside out, right? Because normally what happens is our body is aqueous environment, as you know, because we're made up of primarily water, and the protein will hide all the hydrophobic parts on the inside to stay away from the aqueous environment as much as possible. So if you've changed your hydrophobic environment, all of a sudden it's going to flip inside out. And of course, the enzyme is not going to function anymore. Okay, next one. What is the question D? Question D is, what does the enzyme lactase do? Right, so anything that ends with ASE means it's probably an enzyme. So the enzyme lactase will take lactose. Okay, so lactose in the presence of lactase is going to cut it back into its monosaccharides. What makes up lactose? Anyone? Yep, Rueda said galactose, and glucose is the other one. Very good. Okay. So the lactase will bind to the lactose will bind to the active site of the lactase. Lactase will break it down into these things and release it. Okay, moving on. Complete the following chart. So what are the monomers of carbohydrates? This is definitely a chart that you want to do on your own and write down as much detail as you possibly could. It will definitely help you. So monosaccharide is correct. And there are a few types of monosaccharides that we learned about, right? There is glucose, there is galactose, and then there is also fructose. Okay, so OSG again typically means it's a sugar. Biological functions, we have a bunch. Primary source of energy. Okay, so an example of that would be glucose. It might be storage form of energy. An example of that would be starch in plants and glycogen in animals, where we store our excess glucose as glycogen in the liver and muscle. Christina says structural support. That is correct. Structural support. And an example of that would be cellulose for plant. And the other one that a lot of people kind of overlook is chitin, which is structural support for bugs. Their exoskeleton, their hard shells, right? Same as for like crustacean, which is like seafood with shells, right? Shrimp, lobster, crabs, all that stuff. Okay. Moving on. Proteins. The monomer for proteins are amino acid. You should probably know what an amino acid look like. Again, you do not have to draw it, of course. But if I show you a picture of it, you should recognize, oh, that's an amino acid. There is an amino group here, and then there is a carboxyl group over there. Okay, so this is amino group. over here, we have the carboxylic acid. Okay. Now I'm just gonna put like a note here, just because I remember there might be a question like this. A peptide bond connects an amino group of one amino acid to the carboxyl group of another. Okay, so we did this before in lecture 2. You can go find the notes or something. Well, this you're gonna remove an OH from the you're gonna remove an OH from the carboxylic acid. You're gonna remove the H from the amino group of another amino acid, and you're gonna create a peptide bond. Right? Let me just show you very quickly over here. Right? You take the OH and the H, and then you connect the C and the N, and you create this peptide bond right here. So if I ask you, a peptide bond consists of what two functional groups? The answer would be amino group and carboxyl group. Okay. Please ask questions if you are confused by what I'm saying. Biological function. What are some biological functions for proteins? There are a lot. Okay. So again, give them to me. I'll write it out. Structural support from Mora. Yep. Structural support. So example of that would be like, you know, nails, horns, that kind of stuff. Right? Fuel, that's correct. Okay. So actually amino acids can be converted into intermediates of glycolysis or pyruvate oxidation or Krebs cycle. Right? Depending on which amino acid you're talking about, you can convert them into the different intermediates, and then you feed them into those places. Yeah, a lot other examples I can see there are enzymes, very good. Okay, transport, right, for membrane, yes. Hormones, right? Okay, so an example of hormone would be insulin. Okay, example of an enzyme would be like lactase, for example. We talk about that. All right, very good. Next one, lipids. There's not a lot of like there's no monomers for lipids, right? It's just components. So we can have a glycerol, for example. We can have fatty acids. We can have maybe a phosphate group, right? So these are all components that we can use to build the different types of lipids. And what are some functions of lipids? Anybody? Functions for lipids. Yep. So cell membrane, major component of cell membrane. So we have phospholipid, right, as well as cholesterol, right? Well, cholesterol is not really a major component, but nevertheless, it is a key component of it. Christina said insulate and protect organ. That is very good. Insulate and protect organs. Yeah, signaling is could be. It's not something that we talked about specifically though. Signaling is more like for proteins. Water resistance barrier. Yes, if we're talking about wax, for example, water resistant barriers. Example, wax. Okay. There are other stuff like hormones again. I'm gonna be invading the space here for nucleic acid. Actually, can I put more? Oh, there we go. Oh, wonderful. I'm sure you cannot do that on your paper, but here we go. Hormones. Okay, so some hormones are protein-based. Other hormones are steroid hormone, right? Steroid hormone. Anybody can give me an example of a steroid hormone, please? Okay, and I see storage of energy as well. Very good. I'm still waiting for that example of a steroid hormone. Estrogen, excellent. Progesterone, testosterone, right? Very good. Good, good, good. Moving on. I hope you guys feel ready for this test. Not difficult, I would say, right? It's just like we did most of that already, right, in the quiz. So, you know, you survive the quiz, you should be able to do well on this. There's only one extra topic that you haven't been tested on yet, right? Which is what is it? The metabolism. Okay, you will feel ready. Okay, you still have time. You have 2 more days. All right. The most important thing is to sleep. Sleeping is very important. Next one is, yes, I will post this sheet today. I'll post it within half an hour after we say goodbye. Okay, I promise. Next one is nucleic acid. Okay, so what are the monomers for nucleic acid? It is the nucleotide, right? Okay, nucleotides, which includes sugar. We talked about that already. I'm not going to write it again. There is the sugar, there is the phosphate group, and then there is the nitrogenous base, right? Okay, so next one. What is the next one? Function of nucleic acids. Well, it is the genetic code, right? Genetic code, right? Which is the instruction to make proteins, right? For example. So that is the function of DNA, right? This is the hereditary material, right? Hereditary, how do you spell it? Hereditary material. Okay, so that's also DNA. And then can be used by ribosomes to make proteins. Okay, so that's going to be RNA. All right. Can you post the Beetroot Lab assignment answers? No, unfortunately, I won't be able to do that because, you know, there are those like websites, right? That, you know, Course Heroes or something, and then the whole lab goes up there. I don't want that to happen. But I took it up in lecture 5 at the end. So watch the tutorial and you will find the answers to those questions. There will be like 4 or 5 lab questions. So make sure you actually do review the lab. All right. Okay, very good. Yes, I know the site. I was once a student as well. We didn't have Course Hero back then, but we had, what was it called? Actually, I don't remember, but we had something similar. Okay, like you can buy like past tests or something. Anyway, how is the shape and the structure of a protein stabilized? Okay, so you know, this is talking about the different levels, right? There's the primary, secondary, tertiary, and quaternary, right? So let's just go through that quickly. So we have the primary structure. Okay, so primary structure is talking about the linear sequence, right? The sequence, right? Sequence of amino acid. Okay, so like if you read the amino acid from one end to another, right? That sequence that you read out, that's the primary sequence, primary structure, okay? And what holds it together? Well, I told you before, it is the peptide bond. And peptide bond is a type of covalent bond, okay? A type of covalent bond, sharing of electrons, right? Okay, so these peptide bonds cannot be broken by heat, right? Now listen carefully. If you heat up a protein, if you heat up an enzyme or something, high temperatures, you will affect its secondary, tertiary, and quaternary structure. But once it unfolds into this, like, you know, linear sequence thing, you won't be able to break down the amino acid. Okay, you cannot chop it off. In other words, okay, the peptide bond can only be broken by enzymes, for example, as well as using some other chemical agent called reducing agent. Okay, but we didn't learn that. The key thing is temperature alone will not break down the primary structure. That's something to know. Okay, and then we have the secondary structure. Okay, so this is when the primary sequence is going to coil up in some parts and then form zigzag patterns in another part, right? Okay, so these coils, we call them alpha helix. And these beta zigzag sheets, we call them beta pleated sheets. And they are stabilized by hydrogen bonding, hydrogen bonds of the peptide backbone. Okay. And then this will, so of course, primary leads to secondary, and then secondary will lead to tertiary, and then it will fold up, right? Like into some kind of three-dimensional shape. Is really hard to draw a 3D shape on a 2D surface, but then you will get something like this, like a overall three-dimensional structure, and that is the tertiary structure. Okay, it's talking about the overall three-dimensional conformation, which is just a fancy word for shape, right? Three-dimensional shape of the protein. Okay, so this is stabilized by various interactions, such as hydrophobic interactions. That means things that are hydrophobic tends to come together. There is, ionic bonding, okay, so on and so forth. You can find a bunch of them in the PowerPoint. There's, for example, disulfur bridge. So all these interactions between the side chains, right? As I mentioned in class or before, the tertiary structure is, for most of the proteins, it stops here, right? It doesn't go any further. But sometimes it could go even a level higher to form the quaternary structure. Okay, quaternary structure, which is when you have multiple polypeptides coming together to form a protein complex. Okay, so this is a protein complex with multiple peptides. So you might have one peptide here, another peptide here, and then here, and then here. So individually, each of them has its own primary, secondary, tertiary structure, and then coming together, right, forming this big, giant complex. That is the quaternary structure. Okay, and it's stabilized by the interaction between the individual polypeptide. All right. Any question? Anyone? You sure everybody's happy? Alrighty. That's it for lecture 2. Now we have lecture 3. 1 down, 3 to go, I think. List the major components of cell membranes. Okay. We did this before, but you know, let's go through it again, and we can add more details to it now that we actually have done more in the course. Okay, so these things that I'm drawing, they are phospholipids, right? So of course, this is the phospholipid bilayer. Phospholipid bilayer. Okay, with a hydrophobic core and the hydrophilic heads facing the outside as well as the inside. Now, because of the hydrophobic core here, things that are small and nonpolar will cross easily. Will cross the membrane easily. All right. Now, of course, the phospholipid is not the only thing that is found in the cell membrane. We're going to have some integral protein, right? So there are many examples of integral proteins that we learn. One examples would be these channel proteins, right? Remember? Channel proteins. So if I have something that's like a charged particle, okay, the charged particle would not be able to go through the phospholipid bilayer on its own because of the hydrophobic layer. So the channel protein provides a corridor, a tunnel that will shield the charged particle from the hydrophobic environment, and it allows it to go through. Okay. So what is another type of protein that also performs the same function as a channel protein? It's a different name. Does anybody know what I'm talking about? Yes, integral protein, channel protein is an example of integral protein, but I'm talking about another type of transport protein that belongs to the same category as channel protein. Does anybody know what I'm referring to? Yes, the carrier protein is exactly what I'm looking for. So there is your carrier protein, which is your Pac-Man. Okay, so it will grab on to a particular substance, it will change shape, and then it will open up on the other side and bring it out. Okay, so this is kind of like a revolving door, all right, and this is just like a tunnel for things to go through. All right, so this would be carrier protein. Okay. We can also just have like regular integral proteins that will allow you to attach to cytoskeleton, right? Okay, so this is cytoskeleton. Okay, you might have some that has enzymatic activity, right? Many other options. So let's just put another one here, right? Okay, we can. This could be like an enzyme. Okay, and it's only on one side, so we call it a peripheral protein in this case. Okay, and then you might have like multiple of these, right? So you know, A comes along, it becomes B, and C and D. So it allows a series of chemical reactions to occur rapidly close to the cell membrane. Okay. Other things that I could add to this, we have the amazing cholesterol right here. The more cholesterol you have, the more fluid the membrane will be. Right? Cholesterol. All right. We might have some proteins that have sugar on them, right? That creates the signaling proteins for cell recognition, perhaps, right? And a lot other functions that we can talk about, right? Forming junctions. So please review that accordingly on your own. Next question. Describe the central dogma of biology. Central dogma means central theme, right? So we have the DNA And we're going to be making a copy of the DNA in the form of RNA, which would then be used to create protein. The process of creating a copy of DNA in the form of RNA, we call that transcription. And when you are building your protein from RNA, it's called translation. Okay. So where does it happen? Well, oops. This is our beautiful cell. This is the cytoplasm. Cytoplasm. And we will have the nucleus right here. Okay, so in the nucleus, we have our DNA. Okay, so the DNA is the double helix. And like I said, through transcription, we're going to make a copy of that in the form of RNA. So there are different types of RNA that we'll learn in the second half of the course. This one is called mRNA, messenger RNA. You might also remember that the surface of the cell membrane has a lot of holes in it, right? There's the nuclear envelope that surrounds the nucleus, and then there are a bunch of these nuclear pores, which would then allow the messenger RNA to exit, and then it will go into the cytoplasm. Once it is in the cytoplasm, we are going to have these ribosomes, okay, that will clamp onto it. Okay. Ribosomes has two subunits, the big one and the small one. They clamp onto the messenger RNA, okay, like a hamburger, top bun and bottom bun. And it's going to be reading the instruction on the messenger RNA, and then it will build the protein. Okay, so this is the polypeptide, which will fold into the protein. That is the central dogma of biology. Anybody have any follow-up questions? Okay. Next one, number 10. One of the functions of liver is to remove toxin from blood. You must have seen this question like a couple times already by now. Okay, so let's just go through it. Which two organelles are expected to be abundant in liver cells? Help me out here. What is abundant? Anyone? SER, that is correct. Okay, the smooth endoplasmic reticulum. Okay, so this is responsible for detoxification, right? Breakdown of lipids. Okay. Another one is lysosome. That is also correct. Lysosome. It has a mixture of enzymes, right? For breaking down toxins, macromolecules, right? Old organelles, and even bacteria. If it happens to be a white cell and it ate the bacteria, it's going to be able to break it down. Sakina said peroxisome. That is also an acceptable answer. Very good. Okay, peroxisome. This one uses oxidative reaction, meaning reaction that rips electrons from things, right? Uses oxidative reactions to break down, again, toxins and other molecules. Okay, but it generates H2O2 as a byproduct. Okay, so those are written. Not so much ribosome. Ribosome is more for like protein synthesis alone. Okay, so there you have it. Okay, I know we asked for 2, but we got an extra one, which is always nice. Okay, moving on. Describe the structure of a mitochondrion, state of function of each part. Sure, we can do that. And let's draw our lovely, lovely oval. And we are going to draw the inner mitochondrial membrane, which folds up into these accordion shape, finger-like projections, whatever you want to call them. Okay, so that's the outer membrane. It is the inner membrane. It forms the cristae. That's what it's called. Inside the inner membrane, of course, is the matrix. Okay. And the space between the inner membrane and the outer membrane is our intermembrane space. Intermembrane space. All right. So what is going to be happening? What's the function? Well, let's start with the cristae. Okay, so the cristae is where the electron transport chains are embedded here. Okay. Remember good old electron transport chain? We have the proteins, right? Where we will deposit the high energy electrons. Okay, so you're gonna have like thousands of these things. Okay, so these purple things, they're ETCs. And you are going to use the energy from the electron that was deposited to pump H plus from the matrix into the intermembrane space. Okay, so the function of the intermembrane space is to allow the H plus concentration to build up. Okay, that's actually not just the function of the intermembrane space, it's also the function of the inner membrane, right? You want the inner membrane to have no holes in it so that whatever H plus gradient you build up, it's not going to be able to leak back inside. Okay. And of course, there is a special protein that looks like a doorknob. Looks more like a sliced mushroom to me, but whatever, subjective. And then what happens is the H plus will go through this special proteins. And in doing so, it's going to be able to create ATP, right? That's basically the theme. Anybody remember what this thing is called? Just asking. That is the ATP synthase. That's correct. If you don't like that long name, you can also call it ATPase. Okay. That's it. Oh, of course, I should also mention in the matrix, right? What happens in the matrix? What happens in the matrix is the pyruvate oxidation plus Krebs cycle. Okay, that's all happening in the matrix. Okay. I cannot believe it's an hour already. Why don't we take a 5-minute break, and when we come back, we are going to do more. See you in a bit. Hello, let's keep going. So here's our application questions for you to to think about. I already see three people has submitted their answers. So I'm gonna maybe give it a minute or so and see if anyone else wants to take a crack at this. Okay, it looks like that is it. I only have 10 people who have answered, and I guess we'll leave it at that. Let's take a look at the question. A chemical reaction occurs slowly at 37 degrees. When molecule X is added, the reaction speeds up significantly. Which of the following is a monomer of molecule X? So before we even look at the answers, which could cause confusion, let's think about what this is actually talking about. If a reaction is going slowly and then you add a molecule to it to speed it up, okay, what kind of molecule are we adding? What do we call a molecule that can speed up a chemical reaction? That's right, it is an enzyme. Okay, so we have to establish that the molecule that is in question is actually an enzyme. And then it becomes so much easier, right? What is the monomer of an enzyme? Well, enzymes are proteins. So proteins are made up of amino acids, right? Hopefully you know that. And I told you, even though you don't have to know how to draw them, you should recognize what an amino acid looks like. Okay, so that's why the answer is A. Is that okay? Any clarification that's required? Okay. That is good. Moving on. Describe the endomembrane system. All right, let's do that. So an endomembrane system is a network of membranes that exist within the cytoplasm, and it starts with the nuclear envelope. So let's draw that out. This is our nucleus. And our nucleus is going to be surrounded by the nuclear envelope. Right there. That is the nuclear envelope. And as I mentioned earlier, it is going to have some holes on it. Okay, probably not as big as this, but there they are. These are the nuclear pores. It allows the control of what goes in and what comes out of the nucleus. Right? Things that likes to come out would be, for example, like the ribosomes that is being made, right? Or the mRNA that is being made. Okay. Now, this membrane, this nuclear envelope, is actually going to extend into the cytoplasm. Okay, it's going to form these channels. Okay. So some of these channels are going to have ribosomes on them, which is what we call the rough ER, and then some will not. All right. So this portion, I'm going to say it's the rough endoplasmic reticulum. And what it does is it will work with ribosomes to synthesize, to make, right, and package proteins. All right. Over here, we have our smooth endoplasmic reticulum, and it has everything to do with lipids. So synthesis of lipids. Remember, lipids include things like phospholipids. So if you want to make like extra membrane or something, you're going to need to do the smooth ER. Okay. Steroids, right? Steroid hormones, all that stuff would come from the smooth ER. Of course, it also breaks down. We talk about breaking down, right? Breaks down lipids, not the other macromolecules. The other macromolecule goes to lysosome, right? Breaks down lipids and some toxins. Okay. So over here, we are going to draw a little vesicle. Right? So there's a vesicle that got butts off from the rough ER, and inside you would have some proteins, right? So this is a vesicle with proteins in it. Okay, and the smooth ER would have its own vesicles, but then it would have lipids in it as opposed to proteins. And what's going to happen is this vesicle is going to travel to another component of the endomembrane system. Can anybody tell me what that other component is, please? That's right. It is the Golgi body. And the Golgi body looks like a bunch of pancakes, right? Stacked together. Golgi apparatus or Golgi body, right? And what does it do? It will sort, modify, and repackage proteins. Okay. So the vesicle is going to come and it's going to get passed, down the Golgi body, and then some might get shuffled back. So it goes back and forth a couple of times, and then eventually it is going to be perfect, and it will be repackaged into a new vesicle that would come off like that. Okay, so this is the finished product. Okay. Now in class, we talk about the various possible fate, right, of this vesicle. Like, where is it gonna go? Okay, so help me out here. What is it gonna do? Yeah, Christina, Sita, they say they could become a lysosome. That's right. Become a lysosome. And we talk about what lysosome does already. So we're not going to repeat that. Adriana said, remains in the cytoplasm for later use. That is exactly, I think those are the exact wordings that I used last time. Very good. Remains in the cytoplasm, right, for later use. And what else? What else can it do? What else is it gonna go? Very good, very good. Okay, what else is it gonna? What is another possible destination for this orange thing? Yes, out of the cell, right? Release the contents out of the cell. What is that process called? If you release large amount of stuff out of the cell, what do we call that process? Through blank. What goes into the blank here? Exocytosis. Very good. Okay. And another thing is it could become part of the cell membrane. Okay. So this part might be a little bit confusing. Let me just show you what I mean. Let's say we have our cell membrane here. Okay, and then we will have our vesicle. Okay, and maybe the vesicle, instead of having protein inside like that, maybe it is oops, maybe, oh my god, what is happening? Okay, maybe there is a membrane protein like that, like an integral protein or something, right? So what happens is this thing will go onto the surface membrane and it will fuse with it. Okay, so afterwards, what is gonna look like is this. So this is the old membrane, and then you will have the new vesicle, right? This one, right? Touch it, and then becomes this, and then you will have your integral proteins like that. So that's what it means. All right. So I hope that makes sense. And that is it. That is it for lecture 3. I mean, there's a lot of stuff that we haven't reviewed here, right? For example, you should know about the intermediate filaments, the microtubules, and the actin filaments, what they do, right? How are they different? We also haven't talked about the organelles that are unique for plants, right? Like, give me some examples of organelles that can be found in plants and not animals, right? As well as vice versa, you should know that. There's also things like centrioles, that kind of stuff. So make sure you review that on your own. Yeah, okay, thank you. It was a general question that I just asked, right? But Christina is correct, right? Chloroplast, central vacuole, cell wall, these are things that were found in plant cells, but not animal cells, right? Things like centrioles, right? That will be found in animal cells, but not plant cells. Wonderful. Let's move on to lecture 4. Explain what passive transport is. Provide examples of passive transport. Okay, so in its simplest sense, passive transport is the spontaneous. I mean, it happens on its own. Spontaneous movement of substances from an area of high concentration to an area of low concentration. Okay. This does not require ATP from the cell. Okay? It just happens naturally. Give me some examples, please. Examples of passive transport. Okay, very good. We have simple diffusion. You open up a bottle of perfume or cologne in one corner of the classroom. Soon the whole class will be able to smell it. Simple diffusion. Or just diffusion is fine. Okay. What else? What else? I'm looking for 2 other ones. Yes, facilitated diffusion is correct. Facilitated diffusion, right? Which is going to be transport that involves the help of a membrane protein, right? Things that are charged, things that are too big, that cannot cross the membrane on their own will require the help of either a channel protein or a carrier protein. That is an example of facilitated diffusion. And see this at osmosis, that's right. Okay. So speaking of osmosis, I would like to revisit this question, okay, that a lot of you perhaps had a bit of trouble last time. Okay, remember this question? There's a U-shaped tube. There's a semi-permeable membrane at the bottom, right? Semi-permeable membrane. That is very, very small pores. And these solutes will not be able to cross the membrane, okay? We did this question before, but I just want to make sure that you know how to answer it, all right? So the question is, if you leave this alone and you let the water move around, which way will the water go? Okay, so is it left or right? Okay, will the water rise on the left or will the water rise on the right? Let's do a poll and see if it's a little bit better than the first time we did it. Okay, go ahead. The question is, which way will the water level rise? Okay, is the water rising on the left or will the water be rising on the right? Okay, I have 8 people who've answered. I would like to see more response, please. Okay, yeah, most people got it. The correct answer is, as Beyonce would say, to the left, to the left. That's the answer. Okay, and the reason is because the water will always move towards the side that has more solute, right? It's trying to balance out the concentration, right? So it's more concentrated here, and the red dots can move. So only the water can move. So over time, the water level will rise. And then over here, the water level will drop. All right, make sure you understand this, especially if you haven't seen this question last time, right? On quiz number, whatever it was, 2, right? Anticipate to see something like this. Maybe you will just be very lucky and not see it again. But who knows? It's good to be prepared. Next, describe the differences between various types of bulk transport. So here we go. Bulk transport. All right. So we can either move things in or move things out. If we're moving things out, we call that exocytosis. Okay. Moving large amount of material, right? That's why it's called bulk. And you know, how does exocytosis work? Well, let's try to draw this out again. Let me see if I can do it. All right. So let's say we have a vesicle right here, and then inside we have all these goodies that we want to release from the cell. So it's going to move towards the cell membrane. It's going to touch it and fuse with it. Okay, and eventually the membrane will become part of the. Wait, did I do this already? I feel like I have done this already. Sorry. I'm pretty sure I did this like a minute ago or something like that. But that's how it is going to be released. Okay, so that is the exocytosis. I did, right? Over here, I think. Yeah, okay, similar concept. Okay, maybe not entirely the same. Sorry, sometimes I glitch a little bit when you teach too many class of the same thing, you forget. So the other way, the opposite of exocytosis is endocytosis, right? Endocytosis, bringing things in. There are 3 different types of endocytosis. What are they? What are the 3 different types? Okay, you can be bringing in mostly fluid, so we call that cell drinking, and it is pinocytosis. Okay, so bringing in fluid, bringing in fluid. We can be bringing in solid, that would be phagocytosis. Okay, solid. And the last one is receptor mediated endocytosis. Okay, and it gives it a little bit more control, a little bit more specificity, because in order for you to bring it in, whatever you're bringing in must fit the receptor that is on the surface. Okay. So let's say we have the membrane here, and then we have these Y-shaped receptors. If it's a circle thing, you won't be able to bring that in because it won't fit in the receptor, right? It's only if you have those little triangle stuff, then it will bind to it, right? And then the next thing that will happen is the surrounding area will cave in, right? Along with the little triangle thing you're trying to bring in. And then eventually it will close off. And now you bring it in. Okay, so this is called receptor-mediated endocytosis. It happens, all over the body. Any question about bulk transport before we move on? All right. Next question. It's also something we talked about, but I just want to reiterate it. So no one's going to be surprised if they see a question like this. This relates to what we are talking about in the lab, right? The cell of a plant that grows in a very cold environment was studied. What kind of adaptations do you expect the cell membrane to have? Okay, so the adaptations need to, what do we do? Do we need to increase the fluidity of the membrane for cold environment, or do we need to decrease the fluidity of the membrane when it's very cold? Should we make it more fluid or less fluid? So when it's very cold, things slows down, right? Things become more solid, right? So we don't want that, right? So the opposite of solid is liquid. So the adaptation needs to increase the fluidity. Oh my goodness. Fluidity of the membrane. Okay. And how do we do that? That's right. We need cholesterol, as Sita said. Increase cholesterol. That's a good one. What's another one that we need? What else is going to increase the fluidity? Now, we can't really change the temperature. Like the plant's not going to be able to heat themselves up, but, temperature would definitely affect it. Yeah. Christina said lipid. What about lipid? Do you want to elaborate on that, please? Yeah, you would want to have an increased percent of unsaturated fats, right? Unsaturated fatty acids. So, those phospholipids that you have in the membrane, right? You want to have higher percentage of unsaturated fats, because if you think about saturated fat and unsaturated fat, one of the differences is the saturated fat would be solid at room temperature, right? So, if it gets colder, it will even be more solid. But unsaturated fat, it's liquid, right, at room temperature. So those do not become solid too easily. By having a higher percentage of unsaturated fat, you would be able to maintain the fluidity at a lower temperature. Okay, so those are the adaptations. Opposite would be true, right? If it's like a desert plant or something, okay, don't want too much cholesterol and you want more saturated fat for a plant that lives in very, very hot environment. Okay, next one. How are active transports similar to facilitated diffusion? So they both require the use of membrane proteins. Okay. How are they different? Well, active transport is uses ATP to move from low concentration to high concentration, whereas facilitated diffusion does not require ATP. Okay, and it goes from high to low. Okay, so pretty straightforward. You probably should think about other comparisons, right? Compare between, for example, endocytosis and exocytosis. How are they similar? How are they different? Right? Compare, osmosis and diffusion. Right? Just try different combinations. Make sure you're able to, sort it out in your head. That would be good. All right, that's it for lecture 4. No questions before we move on to number 5? All right. Number 5 is should still be fresh in your head. And you're gonna get more questions from lecture 5 than the other lectures, right, on the test, just because you haven't been tested on it yet. So first question, what is an organic molecule, right? Organic molecule is any molecule that contains carbon, okay? Except for CO2, CO, and CN. Okay, so the people who came up with the rule, they are like, okay, everything has a C, we'll call them organic. But for some reason, they choose to exclude these 3 things. Okay, so if I ask you to select all the organic molecules in the list below, be sure not to select these ones. Okay, so all the macromolecules that we learned about, the proteins, the carbohydrates, so on and so forth, they are all organic molecules. Okay, organic molecules has nothing to do with the concept of organic food. It does not mean it is occurring naturally. It just means it contains carbon. Okay, except for these ones. List all products of glycolysis. Okay, so glycolysis, we're gonna put in like 2 ATP. We're gonna take a glucose. We're gonna get some. We're actually gonna get 4 ADP, 4 P, and then 2 NAD plus. These are not the products. Okay, so these things are these are the reactants, and then you put it in through the process of glycolysis. And what you end up getting is going to be 2 NADH. You're going to have 4 ATP. You're going to have 2 pyruvate. Okay. And that is, well, you're actually going to get like 2 ADP plus P as well from here. Okay. But these are the major products, I would say. Next question. In cellular respiration, glucose is oxidized while oxygen is reduced. Explain what this means. Okay, so this means glucose will release electrons as it is being broken down. The electrons will be captured by energy carrier, such as NAD plus and FAD. The electrons will ultimately be transferred onto oxygen, reducing it, right? That's basically it, right? It's not a direct transfer. It's not like, from glucose directly onto oxygen. Right? It goes through the NAD plus and the FAD, but essentially, right, it is a transfer of electron from glucose indirectly onto oxygen. Okay. What is the role of NADH and FADH2 in cellular respiration? So NADH, now pay attention to the words I'm using, okay? NADH and FADH2 will be oxidized into NAD plus and FAD at the electron transport chain. Okay, so at the ETC, they will release the electron that they have captured. When they are releasing the electron, when they're losing the electron, they are said to be oxidized. Okay, and then the NAD plus and FAD will return to glycolysis, pyruvate oxidation, and Krebs cycle to pick up more electrons. Okay, essentially they're like shuttling surface, right? Picking up electrons from the first 3 steps, putting them at the ETC. What is the significance of adding coenzyme A to pyruvate during pyruvate oxidation? So let me remind you, pyruvate oxidation is the second step of cellular respiration. And there are 3 things that happens there. We will remove carbon dioxide. We will release an electron, which would then be captured by NAD+, becoming NADH. And number 3, we will be adding coenzyme A. Why are we adding coenzyme A? So coenzyme A creates an unstable bond in acetyl-CoA. That's the thing that it becomes, right? That's the thing that the pyruvate will become after adding the coenzyme A, right? Unstable bond in acetyl-CoA such that it becomes energized and will readily react with oxaloacetate in Krebs cycle. Okay, so to put it in plain English, remember after pyruvate oxidation, we're going to go into the Krebs cycle, right? That spins. The first step in the Krebs cycle is taking the acetyl-CoA and combining it with oxaloacetate. Remember, each reaction requires you to supply a little bit of energy, right? The activation energy that we talked about. Right? So we did that in glycolysis. We charge it up with 2 ATP, and it was able to give us some ATP, split the glucose, and then generate some NADH. But after glycolysis, it kind of run out of steams, right? So to speak, it's kind of used up the ATP that we charge it with, right? So it's a little bit reluctant to react further. That's why the coenzyme A will come and it gives it another boost, another charge, so that now it becomes energetic again, it becomes unstable again, and it's going to be ready to react with oxaloacetate in the Krebs cycle. Right, next question. Assume a mitochondrion contains 50 NADH and 19 FADH2. If each of the energy carrier were used, what is the maximum number of ATP that could be produced? Okay, so let me know. Type some answers in the chat box. I want to know what answers. 188 from Sita. Anyone else? Anyone have a different answer, same answer, agree, not agree? All right. It's correct. 188 is the correct answer. It's very simple. 50 NADH, we will multiply by 3. That's going to give us 150 ATP. 19 FADH2, we will multiply by 2, which will give us 38 ATP. And if you add them together, we are going to have 188 ATP under ideal conditions. Okay. Next one, the inner mitochondrial membrane area is about 5 times the area of the outer mitochondrial membrane. What is the purpose of this? Okay. So by forming the foldings, right, it increases the area. So this allows more electron transport chain to be embedded within the cristae, right? More ETC will increase ATP production and efficiency. Okay, you don't want an NADH to be waiting around for the ETC to free up. Okay, by having more options, if this one is not available, you can drop the electron at another one, right? Okay, and so by having a lot of ETCs and fitting it all in the cristae, you increase ATP production and efficiency. Which steps of cellular respiration generate greatest number of ATP? It is the ETC, right? Okay. A majority of ATPs are generated through oxidative phosphorylation. Phosphorylation means, adding a phosphate to something. In this case, we're adding the phosphate to ADP. And the way we're doing it is by harnessing the energy from oxidation reaction, right? By oxidizing the glucose. Right? Remember, only about 11% is generated through substrate level phosphorylation, right? The rest is generated through oxidative phosphorylation. We did number 9 already before. Describe what happens to the H+, right? We generate a gradient and then they come back down through the ATPase. So I'm not going to do that again. The purpose of the final electron acceptor. Okay? The final electron acceptor, of course, is oxygen. Okay, and the purpose is to remove electrons from the ETC, right? Such that an other NADH or FADH2 can deposit more electrons onto it. Okay. If we do not have the final electron acceptor, then the electron will be stuck there. Okay. The next one won't be able to come down. Remember the analogy about the kids at the park? Right? Slide down. Right? If the kid's not going, the next kid might not be able to go, right? Even if the next kid go, and then the next kid go, eventually the whole slide is gonna be full of kids, right? The next one will not be able to come down, right? So it's gonna get clogged up. Basically, this is what happens if we do not have the final electron acceptor. Okay, well, that is it. I wish you all the very best, right? Study hard and, get enough sleep. Read the questions carefully. And the test is going to be on Thursday. And if you have any question between now and then, feel free to email me. I will definitely get back to you as quickly as I can. So thank you for coming, and I will see you all next, next Thursday, right? For the second half of the course. Bye.